Well, first of all, I would like to thank you, the organizers, for giving this opportunity to talk in this venue. And I also thank everyone here for just attending the talk. Today, I'm going to talk about my work on drug synergy. So as most problems in computational biology, the drug synergy one is the challenging one, and it has many dimensions on it. One of the dimensions is the context special aspect of it. So a given drug that synergizes in one disease may not necessarily synergize in another disease. And this doesn't have to be different diseases. This could be like different subtypes of the same cancer. Even if you find your pair of favorite drugs, there is still the dose aspect of it. So the synergy, you can think of it also as a function of the dose. So you have to also optimize the dose. So given the limited amount of resources that we have and not to overfit the data, we did some choices and simplify the problem a little bit. So basically, we choose the MCF7 cell line, mainly because we had a lot of preliminary data on that. And we tried, and using the data, we also fixed the doses. And instead of doing a many drug pairs approach, we choose 3 drug pairs, which is tamoxifen, mefloquine, and vidaferin. And instead of looking different pairs, we just look across different time points. So basically, we had 3 monotherapies, 3 combination therapies, and 1 control, so 7 conditions, and across 6 time points, matching 42 different conditions. And at these different conditions, we measure the RNA sequencing data, as well as for the phenotypic assessment of it, we also have the viability data. And actually, this is one of the drug pairs that we have in here. Basically, in the x-axis, you have the time, and in the y-axis is the viability. And here, the blue and red ones are the curves for the monotherapies. And the black one is the one that is predicted under the null Bliss model, and the pink one is the what we observe in the actual combination therapy. Basically, the higher the difference between the black curve and the pink curve, the more synergistic the drug pair is. And here you also can see the temporal aspect of synergy. Basically, the synergy just shows up after 9 hours, and it continues around that. So using this data that we have, we answered 3 questions. 1, can we discover some mechanism underlying synergy? And second, can we use these insights to predict synergy using the expression of monotherapies alone? And the third question we ask is, how well does our result generalize? So in the next 4 minutes, I will just try to answer these 3 questions. So as I said, we just use excess over bliss to quantify phenotypic synergy. And we also needed a definition for defining molecular synergy. And we tried a lot of things, but at the end, we end up the most simplest one. Basically, we defined a gene to be synergistic if it is only differentially expressed in the combination, but not in any of the monotherapies. And this is showing the results that we have for 2 of the 3 combinations that we have. Here is a Venn diagram of differential expression. You have the monotherapies as well as the combination therapy. And here is the synergistic genes that we, according to our definition. And basically, we tried to see whether there is a correlation between the phenotypic synergy and the molecular synergy we defined. And in the scatter plot, in the x-axis, what we have is the number of synergistic genes. And in the y-axis, we have the phenotypic synergy, which is the excess over bliss. And what we observe is a very good correlation between these 2 types of the synergies that we defined. And next, this is a similar plot to the first one. Instead of in the x-axis, what we have just done is to find the correlation between the expression of the monotherapies. And what we observe is that the more similar the monotherapies are, the more higher the synergy. So basically, what we hypothesize from this 2 plot is that you have enough correlation between the monotherapies, but your expression should be diverse enough to generate the synergistic molecular synergy that we observed. And the third thing we tried is that we just inferred the gene regulator network using the CMAP data, which was also an MCF7 data. And we tried to predict the synergistic activation of transcription factors in the combination therapy using only the transcription activations of the monotherapies. And there was a lot story, but I just condensed in these 2 network motifs that was the most common ones. One is the AND gate. Basically, you have 2 transcription factors that is active only in the monotherapies. Only when they are together, they give rise to a synergistic interaction in the combination therapy, which in turn also activates other transcription factor that is also unique in the combination. The second mechanism is the double down mechanism, which is basically the transcription factor is active in both monotherapies. Only when you give enough dose of them, then you get activated a new synergistic in the combo therapy, which is going to activate new transcription factors. And we quantified how many of the transcriptional synergy we can explain using these motifs. And as also consistent with our observation between the correlation between the monotherapies, 60% of the synergistic transcription factors can be explained this double down mechanism, but 20% of the rest, 20% of the transcription factors is unique to this AND gate that we have, which shows that there should be a great balance between correlation and as well as the new synergy that you observe. So next we tried to algorithmize these observations, and what we did was we just get the monotherapies and we try to predict using machine learning the number of synergistic genes, and also calculated the correlation between monotherapies, and we just combine them in an optimal way to see whether the drug pair is synergistic or not. Next, we try to validate this in a completely independent RIMS challenge data set, which had 91 combinations. And the great thing with this is it had the exact data that we have. Also, we didn't have to run the competitor algorithm to show that we are performing the best, and they are run by the respective teams. And when we see that, we greatly outperform the best performer in that challenge. And it was surprising because the cell line that was used in this challenge is not MCF7, which was completely different, and the results analyzed very well. So I would like to thank everyone from IBM, especially Gustavo, who was my mentor. And I don't have a poster on this topic, but we have another poster, and feel free to come. And if you have any questions both of that and synergy, I'm happy to answer during that poster time. Thank you very much.
Minute talks are some of the